I am going to go through Chapter 8 covering the same material, but in a different style as the authors of your book. Consequently, I would not encourage you to try to correlate all the details of my lecture with the book, and so I'm going to go through my slides in 10-minute lectures and refer to these as lectures 8A through 8, whatever we get to, and at the end of approximately 10 minutes, just switch to a different recording. Enzymes deserve a chapter of their own, even though they are proteins. And we have just spent an entire chapter looking at the functioning of various proteins. The reason is that enzymes perform many functional roles, but not structural roles as many of the proteins that you have just been examining do. The word enzyme means in yeast. The definition that I like to use for enzyme is that it is a highly specific protein catalyst. This is not a perfect definition, as later in the course we will actually run into RNA catalysts. But enzymes are very specific whether they are proteins or not. And on this slide, I give an example of how specific an enzyme can be. Though we have not yet looked at the structures of carbohydrates, these two molecules look these two almost identically looking molecules on the left of the slide are not treated the same way by the enzyme glucokinase. Note that the only difference between these two sugars is that the OH on the right-hand side of the molecule points down in the case of alpha-D-glucose and points up in the case of beta-D-glucose. The original model to explain this enzyme specificity was the lock and key model, and it was proposed at the time when keys look like the key on this slide, which, by having different shapes pass through notches cut in the door, was able to unlock the door. Today, we use a model referred to as induced fit. Induced fit is like putting your hand inside of a glove, making a fist, and having the glove conform to the shape of your fisted hands. And on this figure, what we show is that the presence of glucose, in the presence of glucose, the molecule glucokinase wraps around the substrate such that the blue and orange, or maybe that's yellow, portions of the protein move closer together to each other, towards each other. Many enzymes display stereospecificity, and the polyaffinity mechanism is a way of explaining this. It comes down to the fact that if we have 3 points of attachment or contact between the enzyme and the substrate, then we can bind one stereoisomer but not the other. Enzymes are catalysts which speed up reactions without being consumed in the reaction. They do so by lowering the activation energy of the reaction. Activation energy is the difference in energy between the reactant energy level and the transition state, which is often referred to as being the top of the hill that we have to climb over to get down the other side. The presence of the enzyme lowers the height of the hill, and therefore, at a given energy level, more molecules are able to traverse the hill. There are 6 classes of enzymes. If I discover a new enzyme, what I would do is to send the information about the enzyme specificity that I have to the Enzyme Commission. They would assign a 4-digit number to the enzyme. The first of the numbers, which on this slide I have placed in a light blue color, is the class of enzyme. The second digit is a subclass, which I have in a dark blue. In red, I have the subclass, and in black, I have the specific substrate which is acted upon. The number you would want to keep in mind as biochemistry students is just the first or class number. The first class of enzymes are called oxidoreductases, which are enzymes that catalyze redox reaction. To walk through one example in detail, the subclass of the example I have chosen acts upon a CH-OH group, which is the second carbon from the top of the molecule in this example. The subclass tells us that a molecule of either NAD or NADP is required as an electron acceptor for this enzyme. And finally, the substrate tells us which molecule it is that is acted upon by this oxidoreductase. So in this reaction, what we see is that the OH on the number 2 carbon is being oxidized to a keto group, and the 2 hydrogens, one from OH and one from the H, are transferred onto NAD in order to form a molecule called NADH that we will examine later. I began with this example to make the point that some enzymes need a second molecule by which to act. These are called either cofactors or coenzymes. The cofactor most commonly refers to an inorganic atom, such as iron. Sometimes cofactor means an unknown something, which, if it's unknown, we don't know whether it is inorganic or organic. In contrast, when we see the word coenzyme, we know that it is an organic molecule which is required by the enzyme. Most of the time, the coenzymes are either vitamins or, much more commonly, are vitamin derivatives. I will next go through each of the 6 classes, giving an example of each, but we're coming up on 10 minutes, so I will stop here for this particular lecture.